So like we kind of figured out that drawing stuff out isn't very successful over YouTube. Like the screen recorder I'm using isn't very it's not very good for it. And it could just be I'm using a laptop with a pretty weak graphical processor, so who knows? But either way, we kind of figured out that drawing is not going to go very well using my typical screen recorder. So I'm going to use a different screen recorder and just kind of upload this straight to YouTube. So this isn't live, right? And I just wanted to give some background about comparing sequences, right? Because the whole point of comparing sequences is to estimate relationships among those sequences. And right, a big problem in biology that we've constantly been trying to solve, that we've tried to solve, is what's related to what. And so we first started out using character matrices, like so the presence of a lung or the presence of limbs, the presence of fur, the presence of feathers, et cetera, to try to estimate species relationships. But there's a problem with character matrices and presence absence of specific characters is convergence, right? Just because two animals share a specific trait doesn't necessarily mean that they share a close common ancestor. But DNA sequences, it looks like they don't have, most of the time they don't have that problem. So that's been the go-to way to estimate species and gene relationships, right? So anyways, and so I just wanted to give a little bit more background about that. What's going on behind the scenes in these methods, right? There's a difference between clicking buttons and understanding what we're doing when we click those buttons. So let's start out with a reasonably simple DNA sequences and observe what's kind of happening behind the scenes, at least in a basic manner. So let's take a few reasonably simple sequences. So I'm going to have ACTG. So that's going to be animal A, B, and C. So 3 sequences. B is going to have ATTG, and C is going to have AAGG. All right. And so one of the first things that we're going to do is just estimate how different these sequences are from each other. So first I may compare A to B. So whenever I look at these 2 sequences, we have 4 bases to look at, and only one of them is different. So the second base, C to T, is a difference. And so if we're calculating the overall distance between those 2 sequences, it's basically the number of changes over the total length of the alignment. So this would be 1 change out of 4 bases, or equivalent to 0.25, right? That's a raw distance of 0.25. We can do the same thing, B to C. And here we have 1 change there and another change there. So we have 2 out of 4 changes. So 0.5 is going to be that distance between B and C. And A to C, if we compare A to C, let's see, we have the C to A and a T to G, and those are the differences. So that's also going to be a distance of 0.5. Okay, and so we can kind of draw this out in a neater way using a pairwise distance matrix, where it's going to be like ABC down and ABC across. And so A to A is going to be the same. It's going to be 0. B to B is going to be 0, and C to C is going to be 0. But A to B, this is going to be 0.25. A to C is 0.5, and B to C is 0.5. We could do the same thing on the other side, but they're going to be the same numbers and mirror each other. So there's really no reason to fill out both sides of a pairwise matrix. But right, these are going to be our distances. And even just based off of this, we can see that A and B are more similar to each other, which suggests that they would share a common ancestor more recently than to C. So C is going to be a ancestral lineage, and A and B are more similar to each other. Okay, right, so this is very raw, this is very basic, and not entirely accurate because, right, we have this, not all bases have an equal chance of mutating into another base. Furthermore, we have to take into consideration, just because this fourth position all have a G here, doesn't mean at some time in the past, species A had a C, but then switched back to a G. So we tend to have substitution matrices with specific rates take into account, because one thing that we see when we compare two sequences is just because we observe differences doesn't mean that is actually how different those sequences are. Because if we look at sequences, if we compare like time versus divergence, so let's have an X and Y axis where time is moving forward, and we're comparing sequence divergence on the Y. And so this will be between 0 and 1, because that's the max, is that we can have completely identical sequences or 100% different sequences. And so we can have this kind of rough expectation that as time moves forward, those sequences will differ. As you'd expect, they're independently evolving sequences, but because there are only 4 letters in this alphabet, we have this problem called saturation. So we can have this example where if we actually if we measure the differences between two sequences, it may follow this curve for a while, it may follow the straight line for a while, but then it will kind of plateau out. Even as time moves forward, we'll ultimately reach this point of saturation. So this is called saturation. And this is where we basically can't tell the difference or the distance of these two sequences anymore, how different they actually are, because we only have 4 bases. So even if we have a string of 1,000 bases and they're random strings, 2 random strings that are 1,000 long, 25% of those bases will match just on chance alone. So at some point, we'll reach saturation. It's effectively like any mutation that can take place has already taken place, and we can't accurately measure the distance between these two sequences. Furthermore, sequences that may appear more similar, we've seen additional mutations than what's actually observed. And so we have models of substitution to help correct for this saturation problem and provide a more accurate distance between our sequences. And so the simplest substitution matrix that we've seen, that I've kind of briefly talked about, is Jukes Cantor. And this is the first, or JC69, because that was the year it was derived. And so in this substitution matrix, we account for 2 variables, really. So A, G, C, and T. And so in a Jukes Cantor model, all of these changes are equally likely, right? There's a certain mutation rate that we call alpha that's applied to all base. So we have the substitution probability or substitution rate where all is equal to alpha. And the rate of change for A to C is the same as it is to A to T, is the same as it is to A to G. But also we take into account, the Jukes Cantor also takes account into an A changing into a C, and then at point number 2, C will change back to an A. So what may be reflected as an actual distance may be an underrepresented distance and may be underestimated. So this raw pairwise distance that I actually came up with is probably an underestimation. Because we're not accounting for chances where G changed to a C and then changed back to a G. We're not taking account into, I guess, invisible mutations. And there's also an equal probability there's also a probability that A will stay at a G will stay at G, a T will stay as T, and a C will stay at T. All right, we have this additional complexity of taking into account, just because we've seen, to take into account unseen changes. But this isn't exactly a very good model. It's primitive, and it works to some degree. In some cases, it has been used, right? It's not like we've thought of it and then abandoned it. It has been used over time. But we've also increased the complexity of this model. All right, so the next level of complexity is accounting for transition and transversion rates. And so the model, we named the model after Chimera, who derived this first, and it's Chimera 2 parameter, or K2P. Right, this is simple, but again, it takes into account a transition and a transversion rate. And so we may have one rate for the transitions that may be equal to alpha, and then we may have beta taking into account the transversion rate. Again, with the chance of A staying A, G staying G, C staying C, and T staying T. Okay, now that's the highest level of complexity I can describe these models. But what we can demonstrate is how applying these models changes our estimation of sequence differences or sequence distances. So for example, let's revise this a little bit, our sequences, and let's look at 3 sequences. Let's say again, ACGT, and then AGGT, and ATGT. So that's A, B, and C. All right, these are animals, or these are our sequences that we care about. All right, and so if we do like a raw pairwise, right? Right, just we see one difference between A and B, so that's again going to it's my spot, yeah, 0.25. A to C, or sorry, A to C is going to, again, we'll see one difference, 0.25. And B to C, again, we just see one difference, so again, 0.25. Right, so this is the raw pairwise distance between these 3 sequences. So it looks like they're equally different from each other. But again, we're not accounting for the chance that A went to G and then came back to A in any of these sequences at this first base. We're not taking account to mutations that we didn't see. And so whenever we apply the JC correction, we'll get a more accurate distance between these sequences. So now whenever we redo this calculation, this is under the JC69 model, A to B is going to be calculated as 0.304. This is going to be 0.304, and that's going to be 0.304. Right, so it's at least, so it's under the JC model, our sequences are more diverged than what they appear to be, right? We're taking account into invisible mutations that we're not actually observing. But, right, so our distance increases. And under the K2P model, we'll see, let's look at the numbers under the K2P model. So A, B, C. And so taking into account both a transition and a transversion rate, A to B is going to be 0.317 and also invisible mutations. A to C is going to be 0.347. So that's going to be a greater distance. And B to C is going to be 0.317. And so now under a K2P model, A and C are more diverged now. Because whenever we're looking at this, and when we're looking at this second base, this is the only thing that's changing. Yeah, hold on. All right, and so we see this difference because we're estimating a transition and a transversion rate from the data, and the data, at least in this little silly matrix, we see transitions are rarer than transversion. So C to G is a transversion, G to T is a transversion, but C to T is a transition. So at least in this very simple model, because the transitions are less common, it's proposing that where we observe the transition, there's a greater distance, there's a greater difference in these sequences. Now, that's probably in most cases that's not going to be accurate because we know transitions far outnumber the transversions, but nonetheless, we apply these different rates for transition and transversion, and that's going to change our distances. And so why the distances matter is because as we're building trees, we're also estimating sequences of common ancestors at a point. And we may reflect changes as in terms of a branch length. So let's go back to this original sequence that I have here, ACTG, ATTG, and AAGG, where we actually have real major differences, right? 0.25 and 0.5. So I'm going to come back over here, and whenever I draw my relationships between these three sequences, I'm also going to apply a branch length. So I'm going to draw an unrooted tree. And so in my unrooted tree will resemble something like this, where I have A and B here, and then C is out here. Now my distance from A to B is 0.25. And so at least I know my distance from A to B is shorter than it is from A to C. Now my actual distance, I'm going to divide by 2 to take into account I'm going from A to the common ancestor to B. So this might be 0.125. That's 0.125. And the branch length here will be to the center point, to this node will be 0.25. But nonetheless, these branch lengths reflect that A and B are closer to each other than C. And so this is an unrooted tree. And I'll talk about unrooted trees more so in a second, but if I were to root this tree, I may, whenever I root a tree, I'm saying I kind of like, what's my most ancestral lineage? And I want to root from a common, I guess, what node all sequences are derived from. So if I'm rooting it, I'm probably going to root it, in this case, on the longest branch, the most different branch. So C is going to be the most ancestral, and then A and B are going to pair with each other. Right, again, my distance A to B might be 0.125, 0.125. And then from this point to C will be 0.25 or yeah. Right, so this whole branch length itself will be 0.25. And so we can clearly see that this branch is much longer. And so to get to the common ancestor, so to go to get to the common ancestor of A and B takes fewer changes than to get to the common ancestor of A, B, and C. Right, that may be here, right? And so what we're effectively getting at, this is a very primitive idea of a neighbor joining tree. So what I've done here, deriving a pairwise distance matrix, estimating distances, then calculating how many changes have happened since common ancestry. This is effectively reflecting a very weak and a very primitive example of a neighbor joining algorithm to reconstruct the tree from the data itself. Right, so that's what we're doing whenever we calculate a neighbor joining tree. We estimate distances, pairwise distances, bring the neighbors together, right? Those neighbors are going to clump together, A and B are going to clump together. And then from that point, we may estimate the distances from other sequences, right? It's one of the older algorithms to reconstruct a tree, and we're effectively reconstructing a tree from the data itself. Right, it's still a very valuable method. I think I see it less often in phylogenetic analyses now than I did, and especially for like getting a quick tree. A neighbor joining method works pretty well most of the time. But a problem approaches when we have more than 3 sequences. Right, so 3 sequences are pretty easy to handle, but 4 sequences becomes harder to handle. And so what we'll see is this pattern emerge. So if we have number of taxa, so recognize, I guess before we get into this, let's recognize that if we have if we have number of 4 sequences or 4 specimens, we can observe 3 different unrooted trees. Right? If we're ignoring the data, if we're ignoring pairwise distances, and we just have 4 sequences, A, B, and C, and D, right? That will reflect an unrooted tree. And so from 4 sequences, that's one possible tree that we can build. we can also see and lastly, right, so these are the three possible unrooted trees that we can make, right? Ignoring the data, that's just the possibility of trees. And so for four samples and unrooted trees, this is our tree space. Is the total number of possible trees that can be made. Now, if we try to root a tree, we'll get a different number. So we'll see that so if we have number of trees or number of specimens, number of unrooted trees, and number rooted. If we have four animals or four sequences, we can get 3 unrooted trees or 15 different different rooted trees. And so whenever we root a tree, again, it's effectively picking a branch and saying this is where we're going to root this tree. This is where we're going to fold over to say that A is going to be the most ancestral sequence, and our node of common ancestry is here. Next will be B, and then C and D. Right, so this is a rooted, and that's a rooted tree on A. But these two trees are effectively equivalent. And so generally phylogenetic analyses don't construct rooted trees, but we can certainly root a tree. It's effectively like what's the most ancestral sequence that all the other sequences can be compared to estimate some distance. Anyways, right, so this is how we go from an unrooted tree to a rooted tree. And again, it's usually picking an ancestral state, an ancestral branch that's going to reflect ancestry. But we can also midpoint a root. So we can also root the tree in the middle and basically fold it over so that like A and C are where we clearly fold this over. Right, so that's another possible tree that we can make that's midpoint rooted. Right, so we're just saying this is the ancestral node. Fold this tree over, and this is where sequences are diverging from. All right, so tree space is much bigger whenever we root trees, and this grows fast. This grows really fast. So if we have, say, 8 sequences, the number of possible unrooted trees jumps real quick to about 10,395 possible trees that we can make that of 8 sequences. And rooted trees, that jumps up real fast to like 135,000 different trees. Right, that's just for 8 taxa. And if we jump up to 10 taxa, that will go up to 2 million plus. And if we jump to, say, 22 sequences, well, let's just jump up to 50 sequences. That's going to jump to 3 times 10 to the 74th possible number of trees that could be made of 50 samples. And just so we're clear, this is equivalent to the number of atoms in the universe. And if we have 100 sequences, this will jump up to 2 times 10 to the 182, right? So the more sequences we add, the bigger our tree space becomes, right? This is our entirety of tree space. And whenever we have 100 sequences and we do pairwise comparisons, pairwise distance comparisons, even with the best substitution model, we may not come to the right tree, right? Because we have an enormous number of trees that could be good. And so how we address this problem is effectively this is an optimization problem, right? If we have that many trees are possible and we want to find the best tree, we shift from a neighbor joining to a maximum likelihood. And so maximum likelihood is usually favored it's favored over neighbor joining because in maximum likelihood, we're going to sample that tree space and try to find the best tree in all of that tree space. However, it is impossible to sample this many trees. This would take, even with the best computers today, to sample all of that tree space would take like the entirety of the universe to do, right? It's impossible to do. And like many optimization problems, this is an optimization problem. We take shortcuts, and maximum likelihood effectively takes a shortcut to sample all of that tree space. So if we kind of have an example of all the possible trees and likelihoods of a tree being true, we'll get this three-dimensional landscape, right? So this reflects, right? We have all the possible phylogenetic trees that we can make and how good they are, right? Likelihood you can also substitute with goodness. And so maximum likelihood, it's like we're trying to find the tree that explains the data best, right? The likelihood of the tree that explains the data best. And so this is going to be a tree hill climbing algorithm because some trees are going to be absolute trash, right? So, right. So if we go up to this very simple example, wherever I had this as ABC, so this thing, right? So if we had, right, we have three possible trees, right? A, B, and C. Where this seems to be the best explanation where A and B are paired and C is distant. But we can also have a scenario or a different tree where B is the ancestral and A and C would be sister. And right, and we can also get another tree, but we know given the data, that tree is better than this, right? This is a relatively a crappy tree. And so the likelihood of this tree being accurate is much more so than this tree. And so whenever we have hundreds of samples, because even a tree that has 100 samples in it is a reasonably small matrix, right? It's to the point where we'll get hundreds, if not thousands, sequences aligned to build our trees, right? That's not entirely uncommon to have thousands of sequences in a single alignment. And so some of those trees are going to be absolute crap and some of those trees are going to be pretty good. But what maximum likelihood does is it effectively takes a tree. It's going to say, I'm going to quickly design a tree real quick and estimate how good it is. Okay. And so say that tree has this kind of likelihood, right? It's okay. It's not great. But if we switch some neighbors around, our likelihood increases. Okay. Well, let's switch some branches around again. Okay, our tree increases, it's better. And then we increase our trees, we change some branches again, but what we do is we reach a local optimum, right? Within all of tree space, this tree is pretty good, but our optimal tree is over here. And so this is the tree that we want to find in all of tree space. And so maximum likelihoods algorithms, they'll jump around, they'll kind of sample trees in all this space kind of randomly. And then also ask, okay, if I change neighbors, does that tree get better or worse? If it gets better, keep climbing that hill. Change the neighbors again, see if we can get that tree better. If it gets better, keep doing it until we get to the optimum. Right? Again, because it's impossible to sample the entirety of this tree space. Now, this tree space may be real, right? This may be all of our optimal tree space, but our maximum likelihood algorithm may, right, start here and then climb and then reach an optimal and say, hey, this is the best tree I could find. From what, given my starting point, this is the absolute best tree I could find. So here you go. But it's not the best tree that we could get. And so this is a limitation of maximum likelihood. We may approach an optima, but it may not be the global optima. It may be a local optima. But nonetheless, the goal of a maximum likelihood search is to find that global optima. And so we may run, it's not uncommon for me to rerun a maximum likelihood algorithm several times, just in case one of my starting trees climb to a local optima and I'm missing the true tree or the best tree that can explain the data. And so that's what we happen when we click the maximum likelihood. It takes longer because we're not just building a tree given the data, we're building a tree and asking how good that tree explains the data. And then build another tree. How good does that explain the data? Build another tree. How good does that explain the data, et cetera, et cetera, et cetera. Now, I would say the most sophisticated algorithm for building trees at this point is using Bayesian analyses. And Bayesian analyses is a little bit different than maximum likelihood. We still have this massive amount of tree space. But what's going to happen is we have a starting tree. And we're going to climb this hill until we get to this point in this hill. And then we say, okay, we found this great optima. There's like 1,000 different trees up here. Well, let's just sample all of them. Let's just jump around, take all of them, sample them, and see how often we kind of see the same tree. How often does the same tree appear over and over and over again, or something along those lines? Bayesian's a little bit tricky to explain, but maximum likelihood tries to find the best tree. But Bayesian analyses assume that all the trees at the top of this hill are equally good. So let's sample a bunch of them and compare them to each other to say, okay, well, given the optima, here's the best tree. Given this entirety of trees up here on this hill, here's a consensus among all of those trees that we found. All right. And so last thing I'll mention, or I'll try to mention, is bootstrapping. So maximum likelihood and neighbor joining trees bootstrap the data. And so I'll try to draw this out so we're kind of clear on what's actually happening. So let's have 4 sequences, A, B, C, and D. And so sequence A, I need a few more letters to get this point across. So let's say A, G, T, C, A. And then B, and then C, and then D. And so we have columns in this matrix, right? We have to recognize this is a matrix where we have rows and columns. So that's column 1, 2, 3, 4, 5, and 6. Now, just kind of looking at this tree, we can build what would ultimately be constructed of this tree would probably be a rooted tree where C and D are most similar, A and B are most similar. And that's going to be our tree shape, right? If we root it. Okay, but how robust is this data? So effectively, we can use bootstrapping to test how robust the data is in retrieving this tree. And so how bootstrapping works is by sampling columns. So bootstrapping is going to work by sampling columns at random with replacement for the length of this alignment. So we have 6 positions, and so in our first bootstrap replicate, we have to sample this matrix, columns from this matrix 6 times. So let's see how well I can do this. So let's just 4. Let's see how well, come on, be my friend. My pen is not working. Sorry, I took a pause, so things may be out of place. I paused it because my pen wasn't working, so I needed to fix it. So let's see if that works now. So I'm going to sample this first column. Yay. I'm going to back up a little bit. All right, so I've sampled that first column. So this is going to be my bootstrap replicate 1. And then I may sample a random column. So let me sample column number 5. So that's going to be a random column. And so that's going to go next, right? I just randomly sampled that column. And so that's going into my bootstrap alignment. But I can sample with replacement. So I can certainly sample column number 5 again, right? That's fair because I'm sampling with replacement. And then maybe I'll sample 2. And I have to keep doing this until I get to 6 bases. So I've done 1, 2, 3, 4. Let's do a couple more. We'll get to 4. And sample column 3. Why not? Okay, and so that might be a bootstrap replicate. And so from this bootstrap replicate, then I may redraw my tree again. So I may do my maximum likelihood search again, or my neighbor joining tree again. And it comes out that still, even with this sampling, A and B are different enough. The sequences of A and B are different enough that even if I resample them, they're going to come out sister, and so are C and D. Okay, and so that's just one bootstrap replicate. And in many cases, I'll do that thousands of times, right? So it's not uncommon to see bootstrap replicates like 1,000 times or 10,000 times. Computers are faster, the algorithms are faster. So we may bootstrap a tree 10,000 times to test our data. And so ultimately, if we do this 1,000 times, from our main tree that we drew from the data, right? This would be our primary tree constructed from the entirety of the data. And then we may see percentages at these nodes. So we may see 100% there and 100% there, because no matter how many times we bootstrap this, we kept seeing these sequence relationships in all of our bootstrap replicates. And so this will always usually reflects a percentage. So we're saying 100% of the time among the 10,000 bootstrap replicates we made, A and B were sister in 100% of those replicates. C and D were sister in 100% of those replicates. So we'll see these bootstrap values. And again, it just kind of tests, it tells us how robust the tree is or how robust the data support the tree. And so Bayesian analyses don't do that, but maximum likelihood certainly does. Neighbor joining certainly does. And the whole goal of all of this is to find the simplest tree, right? So evolution and parsimony are certainly linked, right? Finding the simplest explanation of the data. Maximum likelihood, neighbor joining, they all try to find the shortest and the simplest tree that explains the data. But at this point, this is as good as it gets. And phylogenetics is a reasonably solved problem, but like any optimization problem, optimization isn't solved. Because to solve an optimization problem, we would have to sample pretty much every instance to find the optima. But that's impossible to do. There's maximum likelihood, finding a tree, changing some branches around, rebuilding the tree, right? That's taking a shortcut. Like all optimization problems, they have to take into account. They take shortcuts. But nonetheless, so that's about the best I can explain what's happening, going on behind the scenes whenever we click these buttons. But I went on a tangent. I wanted to say that phylogenetics isn't necessarily solved, but it's pretty good because whenever we test these programs out, test these algorithms out, the authors, the researchers use both simulated and actual data. So they may generate a simulated data set that's designed to evolve in a specific fashion. And then we use that tree building algorithm to see if we can reconstruct the evolutionary history that is known, right? So when we take data from nature, we don't know the ancestral state. But when we simulate data, we can create an ancestral state. And so we know the ancestors. We know what those look like. And if our trees approach the evolutionary history, then we know we're on those algorithms are on the right track, right? So they're as good as they can be. Really, I think where the improvements are, and I hope I don't get in trouble, but the improvements are speed, right? Because maximum likelihood takes a very, very long time. It used to take a really long time to do. Now, even with my most complex data set, it may take a few hours to bootstrap it, 1,000 times. We'll just take a few hours to do. Back in the 2000s when I was using POP, that took days. Even a reasonably small data set could take days to complete a maximum likelihood search. But now we're in terms of hours. That's pretty nice. All right, so that's all I need to say there. Hopefully this, I'll talk to you all regular classes, so this will get better. I think we'll see advantages of YouTube when we're on the terminal doing this stuff just with a terminal.